welcome back. I'll start this section for 2.5 days. First of all, I'm Mina Choi, one of the organizers for May Institute. Before we start, I will just reminder or announce several things. We have a Google Drive here. There are all the materials and slides and so on. You can download from over there. And then you can go to the Piazza. We are communicate everything through the Piazza, all the announcement, and if you have a question and so on. So please check the Piazza for all the announcements. And then especially tomorrow, we will have a participant dinner. So please go ahead and then take a call. We need a couple of people who can attend tomorrow. And also around the 4 p.m. we have a poster session. This is not the really official poster, but we just combined all the people together and then just we can write it down. You can make the new poster like very short time and then 1 minute presentation. So we just want to share what kind of interesting you have or your research and so on. That is the announcement. Just general start for the visualization session. We have 4 great speakers for visualization section, and then especially I will introduce our first speakers, Professor Alexander Lex. He is the assistant professor of the computer science at the Scientific Computing and Imaging Institute and the School of Computing at the University of Utah. He directs the visualization design lab and conduct the research on the visualization method and system to help to solve today's data analysis problem in the biomedical science, which is really good matching for our topic. So, welcome, Alex, and then please start your lecture.
Okay, great, thank you. Welcome. So just to get the sense of the room, I'd like to know a little bit about you. Can you just raise your hand if you have some kind of computational background, like bioinformatics or computer science? Okay. Who's mostly from a biology or science background? Okay, great. Anybody who is like design, journalism, or anything like that? No, great. So that means we have a couple of computational people, mostly biology people. So great to know. Okay, so as Mina said, I run the visualization design lab together with Mariah Maya. Mariah Maya is another faculty. She's currently on sabbatical in Vienna, so that means she missed our last group retreat where this picture was taken. And so this is our group. We are postdocs, 2 faculty, some postdocs and mostly PhD students. And we do research on biological data visualization. I would say more generally data visualization, but a lot of applications in biological data visualization. I'm also the co-founder of a startup, datavision.io. DataVision builds visual analytics tools for the pharmaceutical industry. They use that for drug discovery, but also for patent analysis and so on. And I also do teach professional database courses. These are usually 1 or 2 day workshops for corporations. And so if you want to learn more about that, you can go to that link. One other question, who here has taken a data visualization class before? Raise your hands. Nobody, great. So what I'll do today is I will give you kind of like a 25-minute basic introduction into what is data visualization and what are the kind of things that you should think about. And then I talk about a couple of specific research projects so that you get a sense of the type of things that are currently interesting in biological data visualization as a research area. So I'd like to start off my talk with this quote by Richard Hanning. The purpose of computing is insight, not numbers. That's kind of meant in the context of supercomputing. So if you run a lot of compute cycles to learn something about a data set, what you really want at the end is something to learn about what you've done. You don't really care so much about the specific numbers. You care more about what does that mean in the real world. And so if you just modify that quote slightly to say, the purpose of visualization is insight, not pictures, doesn't again sound very controversial, right? I build data visualizations so that I can learn something about the data. However, sometimes I feel like this is not really how it's used in many situations nowadays. And so let me give you an example. Here we have 6 different plant species banana, date, cress, rice, sorghum, and brome. They all have something in common. They're kind of important food plants. And so we want to understand how are they evolutionarily related to kind of get an idea of like anything that applies to one of these plants can also apply to other plants. So how do we do that? One thing we can do is we can look at how many genes do they share. And this was actually what people did in this Nature paper in 2012. I call this the infamous banana Venn diagram. And I would say that this is a terrible data visualization. Who thinks it looks good? I think it looks good. But who thinks it can communicate data well? So that's my problem with it, right? I think it looks fairly good and it makes the paper prettier, but it's not a useful data visualization. And so what this is supposed to show you is the genetic overlap between the banana genome and these five other plant species. And obviously the banana set is the big banana, but everything else are these like regular shapes here. That is that it doesn't like at all encode important items well on the screen. So for example, up here we have a tiny segment with 1,151 shared genes. And right next to it we have one segment with 40 shared genes. So there's no relationship between the prominence on the screen and the prominence in the data. In contrast to that, down here we have a big segment with only 105 shared genes. And also it's kind of tricky to even like see which of these sets participate in any of the intersections in the first place. So you'll have to use a pen and trace the contours to really understand what's going on here. And so this is not good data visualization. In contrast, good data visualization is about making data accessible. It's about combining the strengths of humans and computers, and these are very distinct, right? Like we, as humans, have like a lot of contextual knowledge that the data set that you have on your computer doesn't have, whereas a computer can easily calculate a regression or something like that. And good data visualization is about also enabling insights and about communicating. So some of you might be thinking that, well, I can just like compute my answer, right? I can do some statistics and then I can like describe my data set in terms of statistics. And you're partially right, but you always have to look at your data first. So like this is a famous data set that's called Anscombe's quartet. So we have 4 different data sets here, both with an X and a Y dimension. And the interesting thing about this data set is that all of these 4 data sets have identical means in X and Y, identical variance in X and Y, identical correlation, and identical linear regression. So if we just did some basic descriptive statistics, we would conclude that these data sets are identical. But if we plot them, we see that they're actually very different. So we have like a linear trend here with some noise, a non-linear trend, and then the effect of 2 different outlier configurations. So very obviously, you need to look at the data set before you can make any like, assumptions or summary statistics, just to make sure that what you're expecting is actually there in your data set. It turns out that this isn't just like one particular data set, but you can actually create all of those shapes and all these intermediate shapes and still keep mean, standard deviation, and correlations constant. So you can actually, like this is fake data, right? So you can create like data patterns that don't show up at all in the summary statistics. So I like to think about visualization as part of the data science process, and I like to talk about this particular figure by Kathy Neal and Rachel Schutt that kind of like shows us the data science process. So we have the real world. We collect something about the real world, be it like your Netflix history or some RNA expression data. We process the data, we clean the data, and then here in exploratory data analysis is where visualization is super helpful, right? So like once you have done the processing and the cleaning, you kind of need to look at what is in your data set. Then you do some machine learning, or you build some models, some statistical models, or you run some algorithms, and then you can build a data product that would be kind of like if you want like to recommend the next Netflix movie, or then recommend the next product that you might want to buy on Amazon, then you would build a data product. But whenever you do something a little bit more complicated, you usually want to communicate a data set or some insights to a person. and that person then makes a decision. And so here, again, visualization plays a big role because like communication of data is a big strength of data visualization. I should say that you can also, data visualization also plays an important role in the data cleaning step nowadays. There are like very good tools out there that do kind of like hybrid visualization-based, machine learning-based data cleanup, for example, Trifecta. And also in machine learning, like if you build a model, you very often need to kind of like debug the model. You need to optimize the parameters, and so you kind of like very often use machine like visualizations to aid in that process. But like these two are of course the most prominent examples where you need visualization. So the way we think about visualization in our lab is not really about depicting data to a person, but it's really about human data interaction. It's kind of like a play on like human-computer interaction as its own academic field. We think of visualization as human data interaction. So it turns out that like if you build a visualization, there's a certain like vocabulary that you can use. The question that we want to answer with this is like, how can we effectively encode data using graphical marks, right? And so we have these kind of like marks and channels. So marks are things like a dot or a line or like a cube in 3D. And then we encode data on top of it proportional to some channel. And that channel could be position on a common scale, position on an analyzed scale, length, tilt, area, depth, luminance, saturation, curvature, volume. These are like the non-esoteric ones, even though some are fairly esoteric. And that is like applicable for quantitative data. But right next to it here we have the same channels for categorical attributes. And categorical attributes are very important as well. So we have here spatial region, color, hue, motion, and shape. And the thing is that here those are ranked. So like one of the principles in data visualization is you should think about what is the most important information in your data set and then pick at the top of this list. And then encode the most important piece of data with position on a common scale. And then if you need to add more data on top of it, then you pick from the others here. But I'll go a little bit more into this. And so why are those quantitative channels different? Why do we rank them? So it turns out that there is a relationship between the physical intensity of a stimulus. So like a stimulus, like for example, how big something looks on the screen, how bright I perceive something, but also how heavy an object is would be a physical stimulus, right? And our perceived sensation of it. So we can kind of like, we have some way of quantifying the absolute truth for physical intensity, and then we can also somehow measure the perceived sensation that a human has about the magnitude of a quantity. And so the interesting thing is that the only visual variable that has a linear relationship between physical intensity and perceived sensation here is length. So kind of like scatter plots and the length of a bar, these kinds of things, we perceive them actually absolutely linear to their physical intensity, to how big they are. So there is no kind of like effects, like nonlinear effects if I plot a data point like that. In contrast, if I have something like brightness on a map, I tend to underperceive brightness at the top spectrum. Same for depth and area, we tend to underperceive area. Saturation, we tend to overperceive. And for fun, electric shock, we tend to also overperceive. So this is from a study, Stephen's psychophysical power law. He didn't only study things that are relevant to data visualization, but he also studied things like taste or heaviness and electric shock. And so do not encode data with electric shock because you have like a nonlinear relationship between physical intensity and perceived sensation. So here's this short experiment. How much longer is speed in A? Just like say 2 times? Yeah. Correct. How much longer is speed in A? 4 times, 3 times, 4 times. How much steeper is A than B? A lot of uncertainty, just guess.
3 times.
3 times. It's about 4 times. How How much larger is B than A?
4 times.
4 times, 5 times. It's about 5 times. How much larger is B than A now? This is like 2 times, but it's a bit of a trick question. 2 times by diameter. One problem that you frequently see, especially when people make visualizations with like something like Adobe Illustrator, is that they use the data and just like map it to the diameter or to the radius of the circle. But that you cannot do that. People perceive this as area and not as diameter, right? So like you see that less frequently happen and people actually have to code their visualizations. But if you see like look at infographics on the internet with bubbles, that is a very common problem. So in reality, this is about 4 times by area. So area is like perceived proportional. We perceive the area and not the diameter here. How much darker is B than A? Any guesses?
3 times.
3 times. Yeah, good. So, like, my point here is, notice how everybody got the first 2 right. The bar charts, everybody got them perfectly right, and then there was a lot of like doubt or quietness in the room when I showed the other ones. And so that means that like we're really good at perceiving like the length of the line bar charts, but we have much more trouble by perceiving something like that. So this is of course not the only thing that affects the accuracy of how we can perceive something, but there are other things. Like for example, those are like here I'm using the length of the bars, right? What I just said is like what you should actually use, but it depends a little bit on how, like what else is going on in the visualization. So for example, if we have unframed aligned, this is very effective here. But this here is fairly hard to do, right? So we can even though those are close, we can tell that A is bigger than B, but it's not as precise as in the example on the right. If we add frames to it, we tend to be a little bit better at it. Because it kind of like gives us 2 different things to judge, and kind of like we can compare the white and the blue bar pieces, but it's still not perfect. So other things that affect how accurately we can perceive marks are distractors. So here, like we have these 2 bars are super easy to perceive, whereas these here are slightly more difficult to perceive. Equally, these ones here, and then finally, for those 2 with this distance between them, it's almost impossible for us to tell which one of those is bigger. So it's a little bit more complex and more nuanced in practice, but still, like what I was telling you about position and size being the most important visual variables, visual channels, that still holds. The other thing that is very important to talk about is color. Color is like, if you think about data visualization, then you think about like, how can I use color effectively? The problem with color is that it is, for example, at first it's not ordered, right? Like blue isn't smaller than green, isn't smaller than red. But you do see a lot of these rainbow color maps, and they only really work because you have a legend. And so then you always have to match between like, what does this color mean in the legend and so on. But they have other problems, like, yeah, I'll talk about this maybe a little bit later. But so basically, you should use color only for qualitative data, like categories. You have a very limited number of classes. You should never use more than 7 or 10 colors. Like if you have more than that, you have to like rethink your visualization. It does not work for quantitative data, and there are generally a lot of pitfalls with color. Because, for example, color is very, like we have, like not everybody perceives color the same way. We have about 10% of the male population has a red-green weakness. And so, for example, we should never use any of these red-green heat maps. Like I think the biomedical community has largely moved to these red-blue heat maps, but now, like 10 years ago, red-green heat maps were super common, and that excludes 10% of the male audience, right? And so my rule here is to minimize color use for encoding data and use color just to highlight something on top of it. Let me actually. And so I'll come back to that point. Kind of like at the end of my lecture, I will kind of like give you a couple of examples of how I apply those principles in my own work. The one other thing that is really important in visualization is integrity. So like you've probably heard about like how to lie with statistics, right? So how I can like pick my statistics to kind of make the case that I want, and it's just as easy to do with data visualization. And of course, just as easy to do with the English language. So this is a tool, right? And you can misuse this, abuse it, and kind of like distort what you're trying to say. And so there's a couple of integrity principles on like how to do data visualization. So first, you should show the data variation, not the design variation. So what you're showing should really be in the data, but not based on how you choose your visual encoding. You need to do clear, detailed, and thorough labeling and use appropriate scales. And the size of the graphic effect should be directly proportional to the numerical quantities. These are based on Tufte, and he calls this the lie factor. So let me give you an example of a graphic that I think just doesn't get that right. I'm from Austria originally, and this is from an Austrian tabloid newspaper. This is about like basic income, minimum basic income that people get in Austria, and this tabloid newspaper is like slightly right-wing, I would say. And so they have like here, who gets, in Vienna, who gets this minimal basic income. The first bar here is Austria, then unknown, Turkey, Serbia, Syria, Afghanistan, Poland, Romania, and so on. And so, like, what is the point of this graphic in this newspaper article? What are they trying to do? So the message that you get here is, well, all of these foreigners are taking our money, our taxes, right? They are all getting subsidized. But what's wrong with this graphic? Can anybody spot a problem?
Which is 100 something in the first one. So you basically don't see how much Austria.
Exactly. So you have a label here, but you don't see the bar for Austria because it's just clipped. So the label says 100,000, and then there's like in Turkey there's 9,000. And so if we fix the graphic for them, this is what it would look like. But it doesn't fit on the screen yet. This is the real graphic, right? And of course, it doesn't make their point. And so they very clearly have tried to lie to people here to emphasize an effect that is like very small. And so clearly, this is like they put the number in here, but for the casual reader, they clearly like misstated the effect in the data. So this is clearly a really bad data visualization. So this was like just a very, very short primer. Like I teach a 24 lecture course on this topic. And so if you want to, there's like dataviscourse.net. This is my academic course. You can like download all of these slides and videos, and there's also tutorials for like building web-based visualizations with D3 on there. So like this is, and there's lots of links to books that I would recommend for you to read and so on. Okay, so I want to kind of switch gears and talk a little bit about my data visualization research and give you an overview of like the research areas that I work in, and then I'll talk about 3 topics in more depth. So first, I work a lot in networks. Network visualization, I'm mostly in biological networks. Here are a couple of examples. Here, this is like a tool that we developed for connectivity research so that we can understand connectivity patterns between certain types of neurons. I'm actually showing an example here of connectivity patterns between airports in the United States. So this is like a general purpose tool, but it was developed for this neurology context. And so what's interesting about this tool here is that we don't show only relationships between neighbors, but we kind of like measure connectivity at a certain distance and according to a certain function, and then we depict that. Then here at the top is a pathway visualization tool, like a biological pathway visualization tool that I'll talk a little bit more about. And the idea here is to kind of like visualize these pathways and visualize experimental data like gene expression data and copy number and so on top of it. And then here at the bottom, this is a tool for like path-related tasks in networks. This is in this case a co-author network, but we've also used this for this pathway data set. And so here you kind of query for like the connections between 2 nodes, and then you get a long list of results, and then you can kind of like weigh which of the path you actually care the most about based on attributes in the data set. I'll start off with talking more about this one here. This is about genealogies. And then I do work with multidimensional data a lot. So for example, tabular data and ranking data. And I'll spend some time talking about set visualization. And I saw in your curriculum that you have another lesson on using this particular tool today. I do also work in electronic health records. So this is an example of where we used, where we collaborated with spinal surgeons to build cohorts of the patients that they see for lower back pain, and then help them make decisions which kind of treatment options are ideal. They collect outcome data, and they have a couple of thousand of patients in their history where they have fairly robust outcome data. And now they want to go in and subset their cohorts. For example, if you come in with some kind of slip disc, and you're an 18-year-old high school quarterback, your prognosis and your treatment options are probably very different compared to when you are 65 and clinically obese. And so basically just subsetting these cohorts to have more specific predictions and then look at the outcomes under different treatment options was kind of the objective of this particular project. And then I do some fairly specific genetic stuff. This is an example of where we use TCGA data to the Cancer Genome Atlas to kind of stratify patient populations to help people identify cancer subtypes. The goal of this project was really to take something like clustering, clustered gene expression heat maps, and then juxtapose them with pathways that are overrepresented in any of those clusters, and then look at these effects particularly, but also look at is there a specific mutation that correlates well with one particular cluster, what are the outcomes for these clusters, and so on. Here at the bottom, this is a tool that we built for together with Novartis here in Boston for investigating alternative splicing in mRNA-seq data. And then here at the top is a copy number browser that we are currently building together with ARUP, which is one of the big 4 reference laboratories in the United States. They're kind of switching to a NGS-based diagnostic pipeline, and we're building their copy number browser for that. And then finally, I do work a fair bit now in reproducibility, storytelling, annotation, and integration in computational workflows. Because one of the problems that you have with data visualization is that if you use data visualization, especially interactive data visualization, and then you make decisions, it really doesn't capture your decision process well. If you write a Jupyter notebook or some R Markdown script, all your decisions are baked into the code, right? And you probably have comments and so on. But if you use visualization to do that, all of this information gets lost. And so this is a research project of mine where we try to kind of capture every step in the analysis process and then give people the ability to annotate and to retroactively also kind of curate their analysis processes so that they could say, hey, this is where I made this decision and this is the purpose of that, so that they could share their analysis processes and eventually even potentially capturing people's thought process when they make decisions. And then we can actually create scripts that automatically reapply a visual analysis process to an updated data set. And so that's kind of the idea. This is a 5-year project that just started last year. Okay, so let me get started with this particular project. It's about visualizing clinical data in genealogy graphs. So I'm from Utah, and Utah has a couple of interesting things for this project. So first, Utah, I don't know how much you know about suicide statistics in the United States, but Utah is one of the states that has the highest suicide rates in the country. And that could have many different factors, but one thing that is also unique to Utah, we have this amazing data set that's called the Utah Population Database. And the Utah Population Database is a data set that's based on the records of the Mormon Church. The Mormon Church is very good about keeping genealogical records, and 30 or 40 years ago, they've started to enrich those records with clinical information. And so now we have this amazing resource of going back to the 1800s like where all of these family pedigrees are like stored, and we have information, clinical information about many of those people. Like thousands of ICD values, for example, that are associated with every person. And then so a lot of researchers take this data set and then like run a specific study and study these complex high burden diseases like diabetes or heart disease, or in this case psychiatric diseases like suicide. And so the goal of this study is to really understand the genetic component of suicide. And so we kind of work here with bioinformaticians and psychiatric researchers to kind of study the genetic factors and this genetic risk of suicide, and it's estimated that about 55% of what contributes to suicide is genetic. And so the data set that we use here is we have 118,000 people, 19,000 suicide cases among those 118,000 people. And for these 19,000 suicide cases, we have about 2,000 with genomic data, where we have really like a specific panel, a cycle array for these 2,000 people. And those like 118,000 people are matched into 550 families. And so, of course, you can use, for example, if you have a lot of suicides in one family, you could speculate that there is a genetic component. You could also speculate that they are exposed to the same environmental factors, but that is, like, if you, like you might not actually know your third or fourth cousin, right? And so, like, we know about these big families, and if we see these patterns across these big families, then we can have, we can build a hypothesis that these particular cases are genetic. So let me talk a little bit more about specific goals here. Like, we wanted to find familial cases that also have some kind of interesting phenotypes. An interesting phenotype in the sense of that is probably genetic. So for example, suicide is predominantly male. Like, suicide attempts are predominantly female, but completed suicides are predominantly male. So if you have a family where 80% of the suicide cases are female, then you have a hypothesis that there might be some genetics in play. Or if the suicide cases are associated with rare psychiatric diseases. So for example, it's very common that suicidal people are also depressed, but it's not very common that they also have schizophrenia. If you have a family where there's a lot of schizophrenics and they commit suicide, then this is likely to be genetic. And then, what our collaborators really want to do is to kind of discover these kind of interesting patterns where they have a hypothesis that this is genetic, and then prioritize those cases for the analysis of shared genomic sequences. And so, if you have these 5 suicide cases in a family, you can now take their sequenced genomes and find what are the variants that those people share, because those variants are likely to be somehow involved in the suicide. Of course, you still have to prove that eventually, but this is kind of the process. And then one thing that visualization is very often used for too is proofreading the data. Because as I showed you with Anscombe's quartet, we have all of these kind of weird effects, and that's of course also true for a much more complex data set like that. These data sources are not perfect. We get, when we get these family pedigrees, sometimes they don't even form trees. Sometimes we just get lists, and somebody has just screwed up exporting the expert script or something like that. And so you need to proofread that. And so this is what usually a family pedigree looks like. I'm assuming most of you have seen this before here. We have males here encoded as squares, females as circles, and then this here means that those two people have had a relationship and they have produced offspring. In this case, they've had 5 children, 3 boys, 2 girls, and 2 of them we have information about that they also have somebody whom they had children with. So this is another relationship, and this is a relationship. And then what's commonly done here is that the filled-in symbols here are what's called the phenotype of interest. So in our case, these would be suicide cases, right? So these 2 women and this man here have committed suicide in this pedigree. So that works, that's fine, but the problem with that is this is a very, very small family. And the other thing that we need to do is we need to map all of these data points on top of these people. So we need to know their age, their sex, their race, whether they were bipolar, how often they were diagnosed with bipolar, whether they were depressed, whether they had asthma, obesity, schizophrenia, how they died, which weapon they used, if they committed suicide, and so on. So all of this information and many, many more attributes can be super relevant to the analysis. And so we need to find a way of visualizing this on top of this pedigree. And so like if you take a step back as a computer scientist and think about what is the problem here, it's actually like these pedigrees are graphs. Like more specifically, this is a tree. And we have attributes about those people. And so this is kind of like a multivariate graph visualization problem. And so the question is, how can we deal with graphs that contain rich attribute data? And I'll also circle back to that problem when I talk about the pathways later in this talk. And so people have done this in many different ways, right? They have done things like little pie charts on nodes, or little bubbles, or bar charts, or even like bar charts with error bars and so on. But this is fairly tricky to do because obviously if you have nodes that are as big as this, you will not going to show much of the network. And there are these kind of like occlusion problems and so on. And so like this is kind of like a hard problem in general. And so what I'll show you is a solution to this problem for the specific case of looking at family pedigrees. Here is how like a typical family in our data set would look like. And in this case, visualized with a commercial tool, Progeny, that they use, for example, to create figures for papers. And here we have a genealogy with 400 members. And like, I don't know exactly how many, but it looks like 20 to 30 suicide cases. And then I also show you 4 different attributes about those people. But you can't read them, right? They are like little labels here. So it's just like completely impossible to read the attributes about those people. And so kind of like this is like we have to be better than that to have a successful data visualization. And so this is the tool that we built to do that, and that consists of like 2 main views. So first here we have like a decycled and linearized graph on the left, and then on the right we have an attribute table. And I'll walk you through in detail what the idea behind this is. So first we have like our family pedigrees, they are rooted at a founder couple. So like in practice actually, this is not how family pedigrees look like, right? You have like if you look at your own family, you were kind of like at the bottom, and then you can go back up. But if you go then back down from those, like from your great-grandparents, you will find all your cousins and great-cousins and so on. But if you want to understand genetic relationships, then you can actually just like have a hypothesis that my founder person here has that trait and inherits it to all of his or her offspring. And so then you get a tree, and that makes our life a little bit easier here. It's not a perfect tree though, because people like marry their second or third cousins, and so we have these cycles in here. And so we need to take care of these cycles first. So we kind of decycle this tree, or this like almost tree, to be a real tree by duplicating nodes. So in this case here, I've duplicated the end of the cycle, the node number 7. And if node number 7 had more children, then the children would only be attached to one of those 2. And then this is kind of like the basic idea here. Like you take this tree and then you spread it out so that every node here has its own row. And then by doing that, we can actually juxtapose it with attribute visualizations in a table very easily, like they see here. So this is what this looks like. Like we kind of came up with a slightly modified encoding. So first we position those people by birth year, and then we show like how long those people have lived. So this person here has what we call their age line. This person has committed suicide at the age of 31. He was married, or he had children with this person here, and they had two children, like one female, one male. The male was born first, the female was born later. As far as we know from this chart, both are still alive, but the mother also died very young. So the mother died like a couple of years only after her daughter was born. And so this is what this looks like in the tool. And so you see these like complex pedigrees on the right, and then the big table with these traits, the pedigree on the left, and the table with the complex traits on the right. But one thing that you will observe here is that this isn't a very efficient plot, because we can't show a lot of people. Like I can't show 400 people here, and a lot of the data here is missing. So it turns out that we have clinical data not going back to the 1800s. So we have clinical data for the last 40 years, and we have a lot of data about our suicide cases, but not as much for everybody else. And so we kind of need to do something to make this a little bit more compact so that we can actually see everything that we care about. And so we use aggregation, and that's kind of like a very common principle in visualization. Like if you can't show all of the data, do something like aggregate it in some way, so that the meaningful groupings kind of like pop out. And so this is what we do here. So instead of having, like, we define like people of interest here. Like people of interest are in this case the people who have committed suicide, and then we create this like summary plot where we have everybody who is of interest is guaranteed to have their own row in the table, and everybody else is potentially grouped together with other people. So here we have like others like 1, 2, 7, and 5 share a row. And so to do that, we have kind of like, this is what this would look like in the table then. So here we have like a case, a suicide case. So you see this person is in his or her own row. And you see like this person has died by a gunshot wound. And then we have other people here that are grouped together. And here you see like stacked bar charts to visualize the distribution of those traits. And here like little dot plots. In this case, we're visualizing the BMI. And so you can see that most of those people cluster around like a normal BMI, but there was one person who had a very high BMI. And the one suicide case here also had a very high BMI. Turns out that sometimes we want to be even more aggressive. We have very large families. We only want to show attributes about the people that we actually care the most about, which are in this case, of course, the suicide cases. And so then we use like a more aggressive aggregation approach, which we call like hiding. And so here we show only data for number 6, even though it hides a lot of other elements behind its tree. And then we have occasionally these empty rows. And so like to be able to do this aggressive aggregation, we have to introduce a new encoding for the families. So here we have like the a slightly complex family. We have like one woman who has children with 2 different partners. The first relationship has resulted in 2 boys, and then we had in the second relationship 1 daughter. And that would like, this is kind of like the full-on, more complicated version, and then here we have the compact version where we like summarize this with what we call a family grid. And so here we have this family, and we show you the 2 like men and the 1 woman. They have in total 3 children, and we show all the children here on the right of this line as small little icons, and only the people that are of interest as these big icons that have their own row. That's kind of like a strategy that we use to be able to encode a lot of people. I wanted to, yeah. Here, let me switch to a short live demo. So this is the tool. You can play with it yourself if you like. It's at lineage.kaledoapp.org. You have kind of like a short explanation of what I just talked about here. And so we have a lot of families here, so we can switch between those different families. This is an anonymized subset. Like we have selected 10 families and have anonymized them according to a protocol. We have in our real analysis data set, we have more than 500 families in here. And so you can switch between those families, and then you see like this is now the like compact version. So here I have a family with 80 members, here I have a family with 110 members, and you can see it fits easily on the screen. If I were to kind of like expand that and to be able to see every individual, I do have to scroll a fair bit. But let's go back to the hidden, like to the compact representation. And so now I have like, here are these kind of like traits that I care about. For example, here, bipolar spectrum disorder. I could, for example, sort by that, and now I see all of the bipolar cases. And so kind of like what we see here is that we have a cluster of people at the top that do have a fair number of psychiatric diagnoses, like bipolar spectrum, anxiety, non-trauma, and depression. We can see, like, well, I shouldn't, like, gender is anonymized, so I'm not going to say anything about that here. So the one question that you like now would want to ask is, oh, this seems interesting. Like I care about these bipolar disorders. I could now say, let's start that and find other families where these also occur. And so now I've kind of like, by starting that, I've created this this kind of like metric here that gives us other families where we also have these bipolar cases. And so here we have a fairly large family where we have some of these bipolar cases as well. We can like sort again in the right. Like here we have this large family at the top, and the small family at the bottom, and we see that some of these cases are also bipolar, and we could again sort by that trait, and we see here up here like a cluster of these psychiatric diseases. And so this is how you would kind of like you found something interesting in a particular family, you have a hypothesis of like these traits are associated with genetics, and then you can look at how common is this combination in other families. And then of course, I'm only showing you a couple of attributes here, but you can add many of these other selected diagnostic codes. So for example, where they diagnose as obese and so on to that tool. And so this tool is now being used in this particular study, but we are also like adapting it to other diseases. So for example, we are currently working on getting this getting data for a heart disease study loaded, because like it's very similar, right? Heart disease is very common, and it's a very complex disease with lots of environmental and lifestyle factors, but also with a genetic component. So this is exactly what this is good for. Like the other thing that you could maybe say is that these like pedigree data sets, they're like fairly particular to Utah, but it actually turns out that as we keep collecting data about people and their relationships, these are becoming way more common. So here's like a recent article from Science about a population scale family tree analysis with millions of relatives. It's not based on the Utah data set. So we'll see these kinds of problems pop up more commonly. Yeah, so if you want to like play with this tool, here's the URL again, lineage.kaledoapp.org, or if you want to look at the paper, you can find it on our website at vdl.sci.utah.edu. Before I move on, any questions about this? Okay, if not, let me jump into the a different topic. And of set visualization. And so, like, I showed you the banana Venn diagram, and now I'll show you how we can actually make it better, how we can redesign it so that we can actually visualize sets like that. The banana Venn diagram isn't an isolated incident, unfortunately. We have here, like, the pine genome. This is supposed to be a rat up there. And then here we have, like, worm, mouse, yeast, fly, and so on. And so, you see those kind of like multi-set Venn diagrams pop up frequently in scientific publications. And so like really like when I saw this, I felt like we need to do something better. There wasn't really a good way of visualizing these set relationships. And so one of the problems, like clearly one of the problems that the banana Venn diagram has, it's not area proportional, right? So the segments that are like graphically depicted do not correspond in size to the data that they contain. So you could make an argument that I can just draw area proportional Venn diagrams. So it turns out this is very hard. Like it's actually algorithmically impossible to do it for more than 4 sets, or at least precisely. You can do it heuristically, but not precisely. But it also isn't necessarily a good idea. So here I have a 3-set Venn diagram that I created with Euler Ape, which is a tool that can do area proportional Venn diagrams for 3 sets. But if I take out like the slices of this Venn diagram, which of those two here is bigger, the left or the right? So this goes back to what I said earlier about which kind of visual variables are easy to read for us. And the size of an irregular shape like this is not one of them. So the left one is bigger, and you can probably, like most people, get it right, but it's not easy, right? And so what we wanted to do here is we wanted to create a tool where we used an efficient visual encoding to visualize those intersections, where we also were able to kind of like use interactions to create slices that correspond to some kind of analysis question. For example, here all of the two-way intersections. And we also wanted to visualize attributes about these intersections. Like, for example, is there like something in common about the genes that the banana and the date share? Do they all like have like similar whatever evolutionary age or whatever it is? And so like we built this tool here where we like visualize the intersection in one view, then visualize properties about those intersections in like adjacent to that. Then we have a view about all of the elements where you can also submit queries, and then we can visualize attribute details. And this looks fairly complex right now, so I'll walk you through it in detail. So how can you visualize intersections in the first place? So like the idea that we're following here with Upset is that we take the three sets and instead of encoding it with Venn diagrams, we encode it as a matrix. And by the way, like I don't dislike Venn diagrams. I think they're super easy to read and they're an important tool for teaching set relationships, but they're not a good tool for doing like data visualization of more than three sets. So we have this Venn diagram here, an example with A, B, and C, and then here we are starting to build a matrix. We have three columns, A, B, C. These bars correspond to the size of the sets. And then we use this an encoding here. The cell must and must not, and so it will be obvious immediately. So here, it's not in A, not in B, not in C, so this is everything else that's not in any of those sets. Then here we have in A, not in B, not in C. This is the segment in the Venn diagram up here. So this second row of the matrix here identifies the blue portion in the Venn diagram. And then similarly for just B and just C. And then we get into the pairwise intersections like A and B. So this corresponds to the blue part here. Then we have an AC and a BC intersection. And then finally, we have the intersections of all the three sets. And so now we've kind of like built a matrix where we can easily identify like each of these intersections individually. And note also that I like drew these handles between those cells here. And they're here for a reason, because this matrix is really only meaningful to read in this way, right? Like this is not like the column-wise relationships are not meaningful. What you really want to look at the rows. And this is kind of like what these handles emphasize. And so the nice thing about creating this matrix is that I have actually now have the ability to easily plot a bar chart next to it. So here I can now plot the cardinality, the size of the sets. And so this is the basic idea of Upset. So you have these intersection identified in the rows, and then you have bar charts that show you the magnitude of each of these intersections. And so here it's easy to read, like that this here is slightly bigger than this here, which is not as easy to read in the other chart.
Can I ask a quick question?
Yeah.
So in this plot, it looks like the edges between the nodes in the matrix are not proportional to the size. So that can also be covered.
The edges
Between the nodes and the circles.
So the size is really only encoded here.
Right. So the width of the edge is not proportional to
No. Yeah. It's just like the edges here really just emphasize that like you should read it this way.
Right.
Yeah.
And also one question is, as we can see that for 3 attributes, it's 2 to the power 3. But if we have n, it's 2 to the power n many rows, which grows on exponentially.
Yes. It does grow exponentially, but it turns out that in practice, you're limited by the size of your data set, right? So if you have like, you have 2,000 or 20,000 genes, right? So worst case, if you want to see like pairwise mutation, you would have 20,000 rows, but in practice, that also doesn't happen. So it's exponential growth in the like possible sets, but most of them are empty, and you don't have to show the empty ones. We can now leverage this matrix to also plot attributes about that. So for example, one thing that we need to ask ourselves is how surprising is the size of an intersection? Because let's suppose we have 2 very big sets and then 1 small set. That of course makes a big difference if we have if we just show the size of the intersection. So what we do is we calculate a statistical measure of the deviation of what we would expect of the intersection size to be if it were randomly distributed. Like if we had random set assignment, given the size of A, B, and C, we would expect some kind of like distribution, and here we plot the deviation from that distribution. I'll give you a specific example later, and then we can also plot summaries of attributes about those elements. This is going to make more sense when I show this to you in like a live demo or in a demo. So here I'm showing you a movie genres data set. We have comedy, drama, action, adventure, documentary, horror, and so on. And then here we start out the first. So here we have the movie genres, and then the size of these intersections, then the deviation, and a couple of attributes about them. But so first we have all of these, like only comedy movies, only drama movies, and so on. The first pairwise intersection we have here is the comedy-drama combination, and we see the comedy-drama combination seems to be like one of the largest pairwise intersections, and it actually like shows the deviation measure here shows us that this is a little bit smaller than we would expect. So even though it's the largest pairwise intersection, we would expect more of it, and that's because, as we see here at the top, if we look at this, we see that it's smaller than we expected, but the reason for that is that because comedy and drama are these very big sets in the first place. So other movies like adventures, there's much fewer of them. And then if we look at the attributes here, we see the average rating of drama and comedy here. And so we see that like compared to action comedy, drama-comedy combination has a higher average rating than action comedy. And again, this is not very surprising because action comedies are not usually high-quality movies. So people give them lower ratings. We can then use interaction to reveal interesting facts. So for example, we can ask, for example, what is the biggest intersection? And we can very easily sort by cardinality. We could say, we could do that of course in the tool, sort by cardinality here, sort by deviation, or sort by any of the sets so that we see all of the comedies at the top, all of the dramas, and so on. Or also sort by degree, so all at once, the twos and so on are grouped together. The thing that is then makes this interesting, and again this relates to what I said earlier about like aggregation, we want to kind of like create these groups that are meaningful in some way. So for example, we could ask a question, are many items shared between two sets? And to answer the question, we could simply aggregate by degree. And so here we now introduce a new visual encoding. These are these kind of like group headers. And so first here we have degree 0, then degree 1. And this kind of like corresponds to all of the blue segments. So like this header here, like this is the sum of all of these bars here, of the 3 bars at the bottom. So all the degree 1s. Degree 2 are the green slices here. Again, the bar here is the sum of its children. And then for degree 3, we have only a single one. And of course, the sum of the children of 1 is exactly the same. And so now we can kind of like make judgments about these more complex slices. And so this is how that looks like in the tool. We can switch between different aggregation methods. For example, aggregating by degree, aggregating by sets. So here are all the comedies now grouped. And what we did here is we get these big bars, and so what we do here is we have these bars that wrap around up to 3 times so that we can actually deal with these different scales. So here you see the bar that's wrapped around like, to encode the 1,200 movies. We can also do things like multi-level aggregation. So here I've created overlaps. So now I have all the comedy dramas, all the comedy actions, and so on grouped together. And then finally, we can do more specific queries. Like very often, querying is a very important operation. So for example, we can say, I want to like look at all of the elements that are in A, are in B, but may be in C. So I don't care whether they are in C or not. So I can formulate it like a visual query like this. And this would be the result here. So I have these two intersection match that, and so I've basically created this group artificially. And so here is an example how that works in the tool. You have like this, I've chosen the intersections of sets action and comedy, but excluding the set adventure, and everybody else is I don't care. And this would be the result of that particular query. So these things are really important, mostly in context if we look at elements and attributes. It kind of gives us a more holistic analysis. And so now I'll walk you through like one analysis example for this tool. So I could ask a question, how do documentaries compare to adventure movies? And that's what we'll look at now. So first, what we see here at the bottom is that the documentaries, they tend to not intersect much with other genres. So there's not a lot of action documentaries or children's documentaries, right? We see mostly it's documentaries. There's one or four comedy documentaries in there. And then we have actually, like, if we want to look at the specific documentaries, we have this list on here. But then at the top here, I'm plotting release date versus average rating. And what we see is that there's not a lot of historical documentaries here on the left. And that people tend to like those documentaries, so they give them a fairly high rating on average. So we see kind of like a concentration. Recent and highly rated movies are like documentaries. If we look at adventure in contrast, we can see first adventure mixes a lot. So adventure is, there's like action adventure, children adventure, adventure, action, comedy, all of these combinations. So this is like a fairly common genre to mix. And then if we look at the ratings over time, we can see that, as we already said, like adventure movies are not necessarily like highly rated. So we can see here like a fairly wide dispersion of the orange dots. We can see that there's very few that are highly rated, and most of them are like somewhere in the 1.5 to 3.5 category. But what's interesting is we see historical adventure movies, but we don't see bad historical adventure movies. So what would you conclude is the reason for that? Do you think that nobody made bad adventure movies back in the 1930s? It's probably a data collection bias, right? This is a data set that was collected in the internet age by us rating movies, and we have forgotten about all the bad documentaries from the 1930s. Only the good ones remain. So we have like a bias for historical data. We only remember the things that are noteworthy, and that's why we have very few of those dots. Like we have this this tail here where we have only good movies in history, and then like since the data was collected in real time, we have much more dispersed ratings. And so like you will find this for any kind of like data set like that. But it's kind of an important lesson, because very often we need to like think about how was the data collected before you make conclusions that like previous generations were much better at making movies than us. That would be the wrong conclusion here. So like I've talked a lot about movies, and you guys are here to learn about molecular biology in some way. So how does that apply? So well, it turns out that this can be like used very widely. It can be used in genetics, in economics, in pharmacology, in social network analysis. Here I'm showing a case study we did with a variant calling algorithm, like evaluating 4 different variant calling algorithms and which kinds of calls they make consistently. We also built an R version of this tool. Which was developed by Neil Scalenborg and myself and one of his students, where we made some kind of design adaptations, because like the interactive tool that I showed you mostly here is used as a web page, and so you can scroll, but the R version is usually made for paper figures, so we flipped everything over because that works better for paper figures. And then we have different ways of visualizing attributes. And so you can actually, like in R, this is a package that's fairly popular that you can just like load and make these into publication quality graphs here. And there is an R shiny version. And so now, like to close the loop, this is how the banana Venn diagram looks with upset R. And so like one thing that like you can't tell from that figure, but you can immediately tell from this figure here, the biggest intersection is the one that has everybody, like all of the plants, to share more genes than they have apart. So the biggest intersection is like these 7,674 shared genes between all of those plants. So you can say they are clearly quite related evolutionarily. And the second biggest section here is between the bottom 3 here. The third biggest one is between the bottom 5 here. And the first one, the date, actually does not participate in any intersections up to here. And so we can kind of reason here that the date is probably the least evolutionarily related to all of these other plants. And so, like, might not look quite as attractive as the banana Venn diagram in your paper, but you can actually read something from it, and you can learn something from it, and you can reason about what is going on, and that's the purpose of this particular visualization technique. So here's the Shiny app. So if you don't want to code R, but want to still create the R figure, you can do that with the Shiny app. And my biggest success with this tool was that the guys who published the original banana Venn diagram wrote a follow-up paper just recently, and I was very excited to see that they actually used an upset plot instead of a Venn diagram. So that was a big success story for me personally. Okay, so if you want to play with it here, Toledo.org/tool/upset is information to all the versions. There's also a Python version, which is not developed by us, and there are 2 different Python versions, which is not developed by us, but we've used them and we show them. And there's the R version and there's the JavaScript version, and we also have a redesign of the JavaScript version that's going to be released soon. That allows you to embed it in your blogs. Also, that allows you to integrate the interactive upset tool into a more complex visualization tool easily. So it's kind of like a library, and it does like provenance tracking and undo and so on. And so stay tuned if you're interested in that. Okay, so I want to talk about one last project about pathway visualization. Any questions about the set visualization piece? Great, so pathways then. The thing is with pathways, like you've probably all used PEG or any kind of other metabolic networks in the past, and the thing is that these are really like canonical representations, but don't really account for variation that you find in data between different samples of patients. So you have variation, like for example, I might have a mutated gene, and that completely shuts off a pathway. Branches can, for example, be inactivated to a mutation or to change gene expression or to like desired effect due to drug treatment. So it can shut off particularly one gene by like targeted drug treatment. And so I want to understand that in the context of these pathways. And so this is like a schematic representation of a pathway. And what we want to really to understand whether this pathway applies for everything in your data set is to map the experimental data on top of the pathway. And so here we would have 2 different data sets, one being ordinal, the other one being numerical, and we really want to map both of like these data sets onto that network so that we can study the experimental data at the same time with the topology of the network. And so how can we visualize this experimental data on top of these pathways? And so what people usually do is what I call good old color coding. So we have a vector here with A to F, and we can map it with color. And so that's a good solution, but that works for one sample, right? And for one sample, this is probably the best solution. But if you have more samples, then you have to worry a little bit more about that. So how would I encode these 4 additional attributes here? People have tried with little color bars on top of these pathways, but that's not a scalable solution. So we really want to support these multiple tasks. We want to explore the topology of pathways and also explore the attributes of the nodes, the experimental data that we map on them, and we need to really support both simultaneously. And so the idea that we had here is to kind of like let people get people involved and ask them questions about what they're most interested in, and then show them an idealized version of it. So here we start out with a pathway view, and then we let a person, like an analyst, select, hey, I really want to study the cascade from A to E, and then we extract that path, and we try to preserve as much of the topology. Like for example, here you see that B points to A. We still have that here. That here from this metabolite something goes to C, and then here this metabolite goes into E, and that goes to F, and so on. And then now we can plot the experimental data right next to it. So that's kind of like the basic idea. And let me, you can then also juxtapose this with non-genetic data sets. So for example, like clinical variables, if you have like cancer data, you can like plot tumor staging on top of this, or gender, or anything else that, or like has received a certain medication or not, whatever you like. And so this is how this looks like in this tool. You select a sequence, and then the tool will extract that for you, and then show you this rich like data. In this case, on the left is copy number data, on the right is gene expression data. This is CCLE data, Cancer Cell Line Encyclopedia, from the Novartis collaboration with the Broad Institute. And then you can extend that even like to another pathway, and keep extending your path here, and look at the experimental data for this long path. And so here's how this looks like in the tool. Here you have like a list, a searchable list of all the pathways in KEGG. You select one particular path, and you can notice it on the right here, it is being extracted. And then if I want to look at the experimental data for these, I can simply pop that up and look at these experimental data sets. And so I can make a fairly complicated configuration of these experimental setups here. So here I have two different data sets grouped by non-mutated and mutated. I have like an aggregated representation versus a non-aggregated representation here. And they are sorted, the columns here are sorted by mutations in EG, or actually, by mutations in EGFR. And then now we can kind of make judgments about, do mutations in EGFR co-occur with anything else in this sequence? And then there's also like categorical or other attributes overlaid on top. Here we have gender, days to death, so survival and race overlaid on top of these genetic dimensions. And so, like, why do you care about this? Here's a particular example that we found together with our collaborators at Novartis. So here is an ARP-B signaling pathway, which is fairly important in cancer. And so our collaborator here has selected this cascade here, and then we've juxtaposed it with drug responses to two cell lines. So here at the top we have erlotinib and lapatinib, which are commercial cancer drugs, and we plot here IC50 values. And so basically this tells us, like a low bar here tells us that a cell line responds to the drug treatment. And so what we see is that we have a very differential profile in terms of some of the cell lines really respond well to lapatinib, but the majority doesn't. And so can we see that? Can we find a reason why that is the case in the genetic data? And so, like you would say, well, it looks like ARP-B2 mutate, like an increased expression of ARP-B2 and an increased copy number of ARP-B2 seems to be correlated with the lapatinib being effective. So that would be one conclusion that you could draw. But then you would see that there's two elements here, the gold and the orange one, two cell lines that also have a high expression in ARP-B2, but they don't respond at all to lapatinib. And so why is that? You could just say, oh, outlier, I don't know. But if you use a pathway tool like that, you can like reason about network effects. And so what is actually going on is that downstream in this important pathway, in HRAS, they are shut off, these two. Cell lines, they have a very low expression downstream in that pathway. And so this is like very likely the reason why the drug that is effective for everybody else who has a high expression in ARP-B2 is not effective for those because they have a very low expression in NRAS. And so you can, like with this tool, you can study these kinds of networks effects. And really, like, if you see any effects for one particular gene, and there's outliers, you can, for example, like, use a tool like this to study that. So there's more information about this particular tool. This is still a desktop tool. Everything else I've shown so far was web-based. Okay, so in the last like 5 minutes, and then I'll leave 10 minutes for questions or discussion, I want to kind of reflect a little bit more broadly on visualization design strategies that I use, so that like we can talk about like if you build your own visualizations, like what you should, what of those you should actually also employ. So here are my 5 main strategies, like encoding channel primacy, show relationships explicitly, use queries, use color sparingly, enable annotations and provenance. So let's walk through them. So encoding channel primacy is, and I said this before, but I'm going to say it again because it's very important. The most important data is assigned the most powerful encoding channel, which is position. And so here, like, set visualization doesn't, like the Venn diagrams don't do that, but instead, if we have like the size and position encoding here in the banana chart, that's kind of like not using an ideal encoding and translating it in this principle is what led us to this particular chart here. Show relationships explicitly. So what people commonly do is if they have 2 different views, you select something in one view and then you highlight in the other view. And that's okay, but it's much better to actually visualize relationship explicitly. You can use smart layouts or connectivity to do that. And so what do I mean by that is I could have like just shown this in a table or in something else, some multidimensional data visualization technique, and then shown those people separately in a pedigree drawing. But what we've done here is really we've made sure that we created layouts that allows us to really align everything here, and that makes it super easy to see the relationships between the elements in this pedigree and the attributes about those people. This is the same idea that it also is an upset. So instead of having the like these complex shapes that are somewhere in space, and then visualizing attributes about those movies or about the genes in a separate view, we kind of like made sure that we have a layout where we can show more information about each of these intersections in separate, like in line with them, so that it's really easy to read. The third principle here is use queries. Like I think that's like in visualization, like historically people have always argued for show an overview first, so that you get a sense of every like data point. And I think that's sensible if you have a smallish data, but in practice, it doesn't really work anymore for modern data. Like you use this approach, use queries every day when you go to Google. You search for something, right? You don't want to start with a visualization of the internet and then zoom down into something, but instead you want to use a query to kind of filter it down to something specific that you care about. And so in the stuff that I showed today, like this is one example of how we do that by allowing people to create those queries to kind of make subset this to a data to the data items that you really care the most about. The fourth rule that I use is to use color sparingly. Limit the use to encode data and use color to highlight items of interest. And that you can leverage the pop-out effect. If you have like a mostly gray image and then want to highlight something with color, people will immediately see that, and they will pay attention to that. And so, for example, here in the Lineage tool, we have like color doesn't really include, like there's only one single element that's encoded with color. This is the primary attribute, the suicide in blue. And then highlights are like emphasize like items that I care about. So for example, here I've highlighted this parent-child relationship, and everybody immediately recognized what I'm talking about. And then here, I didn't show that before, but if I care about another variable a lot, I can actually color code it as well. So here I'm color coding a deceased, yes, no, bar, like both here in the columns and next to those glyphs. And then fifth is enable annotation and provenance. What did you see or think when looking at this visualization, and how did I get there, and can I go back? And this, of course, applies mostly to interactive visualization. And that's fairly challenging to do. And even though I think it's very important, it's difficult to do in practice, but we are building this library and framework that allows you to do that. So for here, for example, I'm showing this cancer subtype analysis tool where you can then like add annotations and really explain it and then replay it and so on. Okay, so this is it. This concludes my talk. I'm happy to take any questions. I'll also be around today if you want to like talk during a break. Thank you.